Telomeres have gotten a lot of press because of their possible link to aging. But what are telomeres, and why are they important for human health? To understand this, let's consider how each of us began life, as a single cell. That one cell eventually became the trillions of cells of your body now by dividing countless times, from 1 cell to 2 cells to 4 cells and so on. But before each division, your DNA, which contains the information necessary to keep you alive, needed to be copied so that both daughter cells received the information. But this copying process has its challenges. Our DNA is packaged into several long, thin structures called chromosomes. Well, it turns out that there's always a little bit of DNA at the very ends of chromosomes that doesn't get copied. As a result, with each cell division, there's a loss of DNA. If cells continue to divide, wouldn't they eventually lose important information contained in the DNA? Yes, they would, but telomeres help prevent this. Telomeres are sequences of extra DNA at chromosome ends that do not encode information. Basically, instead of important information getting eroded with each cell division, the telomeres get eroded. The focus of my research is a group of 6 proteins that form a complex at telomeres called shelterin. This complex prevents erosion of important information by bringing in an enzyme called telomerase, which maintains the length of telomeres. Telomerase isn't actually working in most of our cells, except for cells that need to constantly keep dividing, like the stem cells in our bone marrow that make new blood cells. Unfortunately, many cancer cells also have telomerase, which allows them to keep dividing uncontrollably. Shelterin also protects your DNA by making sure your cells don't incorrectly think that the ends of your chromosomes are sites of DNA damage. I am particularly interested in 2 shelterin proteins, which I will call protein X and protein Y, and how these 2 proteins interact. Protein Y has several structural units, called domains, which serve various functions, such as binding other proteins in the shelterin complex. Basically, I and other researchers are making forms of protein Y that are each missing one of these domains, and testing how these different forms of protein Y interact with protein X. If a domain is important for X and Y to interact, then we expect that taking that domain away from protein Y will interfere with the interaction. Though the significance of this interaction is unknown, through this investigation, we hope to learn more about how telomeres and shelterin relate to cancer and cancer treatment.